this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I'm a marketer in life sciences. And today, before we hop into the interview with John Houston, I just wanted to play a quick message from our sponsor.
This episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more.
In this episode of Life Science Success, my guest is John Houston. He's the Chairperson, Chief Executive Officer, and President of Arvinas. With over 3 decades of experience in drug discovery and development, Dr. Houston brings valuable insights to pioneering protein degradation therapies and the challenges of leading a cutting-edge biotech company. So with that, welcome, John.
Yeah, great to meet you. Thank you.
Yeah, great to meet you as well. So would you mind just telling the listeners a little bit about yourself?
Absolutely. Yeah, John Houston, I'm the CEO at Arvinas. I joined Arvinas 2017. The company was just about 3.5 years old, preclinical, private company, had a lot of great technology. I was convinced, having talked to Craig Cruz, who was the founder, and Tim Shannon, who was also the founding investor, that this technology really had a future. So I was very excited about that move, and partly because I had spent the previous nearly 30 years in big pharma, 19 years at Bristol Myers Squibb as the Senior VP in charge of disease sciences and technology platforms up in Connecticut. And then prior to that, 10 years at Glaxo, in the UK, in London. So the transition into biotech was exciting and feeling very different from the big pharma experience. But I've enjoyed the last nearly 8 years now at Arvinas.
It's got to be a big shift, though, to go from big pharma to where you are currently, right?
Well, it was. I mean, I remember Tim Shannon telling me that in his experience, people had moved from senior positions in big pharma into those kind of small, relatively early biotech companies. They either enjoyed getting back to the roots, basically nothing's handed to you. It's, you've got to fix up your own computer, you've got to solve problems, or you just can't deal with it. You've had 30 years in big pharma, things handed to you, things get fixed. And the thought of trying to fix things yourself is too much. I actually enjoyed it. I loved the fact that I was getting data every single day at big pharma. Through governance processes, you get data, but it's removed by several months from the time it was generated. And here I was getting data again on the same day it was coming out. It was absolutely really exciting.
And I would imagine you're a lot closer now to the core of where everything's happening, right? So you don't have, a bunch of layers that then, sort of roll up to you. Essentially you're right at the point where everything's happening.
Yeah, I mean, when I joined the company, Arvinas, I was about employee number 40. So yeah, really very much in the weeds. Now we're, somewhat bigger. We're over 400 people now. But I can still get my arms around most of the programs. Certainly all the people that are based here in the Connecticut site, you can get to see and talk to regularly. But we do have people now, because of COVID, who've been employed who are not based in Connecticut or New Haven. And in fact, we've got staff now, and I think about 25 or 26 of the 50 states, and our head of regulatory actually lives in England. So it has been a broadening out over the last 8 years. But what we have tried to do is stay away from the deadening effects of bureaucracy and governance as best we can. In a small biotech company, you do have the ability to react to data quickly and make decisions quickly. And we're trying to maintain that even as we get slightly bigger.
Yeah, and it's funny because I work with both larger pharma companies as well as a lot of smaller ones as well. And what's interesting to me is some of the companies that have sort of totally embraced this idea of, hey, we have to be in the office. And then there are other companies that have said, well, I mean, we could be a little bit more flexible. Maybe it's, a couple of days a week. And then there, lastly, a third category, which has said, hey, look, we really don't care where people are. We're going to go get the best talent no matter where they're at, around the world. So, yeah.
Yeah, we very much took the approach of finding the talent where it was, and we were able to grow our organization and get great science and clinical leaders in, probably in a way that we wouldn't have got if we'd said, hey, got to move all your families to Connecticut. So it really worked out well for us. And we do have this mixture now of people who are on site every day here in Connecticut, hybrid people that come in and out, and then remote, where they'll see us maybe 2 or 3 times a year, especially when we have our big summer get together, which is always fabulous, getting everybody together. And it's the first chance everybody gets a chance to see each other physically. So that's always good fun.
Excellent. So with all of your experience at BMS, especially with over 200 compounds, in your background, how has that impacted what you're focusing on at Arvinas, and how has it helped you?
Yeah, the experience at BMS was phenomenal for me. As part of the senior leadership team there, getting to see how decisions were made, how programs were progressed, the key issues related to both research and development programs was, a really superb education. And of course, in the role I had at BMS, I was very research focused. I did have exposure to early development and even into commercial through the neuroscience organization there, but largely was research focused. So the transition into Arvinas, where it was all research, meant that I had a kind of a direct connection. I knew and was very comfortable with all the aspects of what you need to do to move a program from point A to point B in research. But obviously, over the last few years, we've done more than that, move our programs forward into pivotal studies, phase 3, starting the beginnings of a potential commercial organization. So the lessons I learned, even tangentially at BMS, are actually coming into play now at Arvinas as I see how our development organization grows out, our regulatory, medical affairs, and commercial organization. So it's been a kind of a truly fascinating journey for me, seeing that kind of arc of getting exposed to it somewhat at BMS, but now seeing it really firsthand and seeing how these teams grow.
Yeah, and the one thing I take from my corporate experience is just this understanding of what good looks like, and then just knowing that hey, there are systems and structures in place for a reason. And so, but I also kind of like the flexibility and agility that smaller companies have. And, I feel like sometimes, there has to be a balance of both. Have you sort of found that? And are you, trying to just continually balance, the need for systems and structure to grow, but also, being able to be nimble at the same time?
Yeah, the one of the biggest differences I found when I came to Arvinas was scientists would come up to me and say, look what I did, look at this great data. Whereas in the big pharma setting, it was, can I do this? So a subtle change, a subtle difference in terms of accountability and initiative. And we've tried to maintain that here at Arvinas, where people don't have to seek permission to do things. The scientists are in the best position to make those decisions, not some senior management individual. So we've tried to maintain that core focus of getting the best people with the right focus to make decisions on programs and science. And that's allowed us to be flexible. It's allowed a good array of targets to be developed. and the technology to be developed further from the days it was when Craig founded the company. So yeah, I do see that as a significant difference from the big pharma setting, where there is a lot more structure, less fluidity in terms of being able to work on the periphery of things. You might have a great idea, but it doesn't quite fit in to what the company wants to do. Whereas we've allowed some flexibility where people can work on ideas and concepts, even if it isn't necessarily core to their original program, because I think that's where you get innovation. You get innovation from those kind of ideas and give them some oxygen to flourish and to grow.
And it's so important for the company, but also for the individuals to just say, hey, look, I feel like I can have a broader impact if I do this other thing, in addition to the main thing that I would normally be working on. So I think you're absolutely spot on. So whenever you look at Arvinas, tell me a little bit more about what it is that you're working on and why is it significant in the field of drug discovery?
Yeah, so Arvinas was the first company founded in the space of targeted protein degradation. As I mentioned, Craig Crews was the founder, and he had spent a number of years working on understanding the protein degradation system in cells, looking at ways of actually initially blocking it, and came off the Caprolus as a drug, along with some of his colleagues, where it blocked the proteasome, which is basically the garbage disposal unit for proteins in your cells. Then he decided there must be a better way, a more kind of targeted way of hijacking that proteasome ubiquitin system. So developing heterobifunctional molecules, one end of which has a ligand that recruits an E3 ligase, part of that ubiquitin proteasome system, and the other ligand binding to a target that you specifically wanted to degrade. And of course, there's a linker. Although the linker part is not minor, it's a significant part of the properties of these molecules. And Craig defined the name PROTAC for them, proteolysis targeting chimeras. And so the initial part of the company's focus was seeing whether or not these molecules truly could pull in the ligase machinery to a protein and degrade it. And for the first 2 or 3 years, that's what the company did. The whole array of different proteins where they made molecules to those proteins and showed that they could actually get them degraded. So very, very exciting. And certainly when I joined the company, the stage where they were at was working particularly on 2 programs, the androgen receptor and the estrogen receptor, one for prostate cancer and one for breast cancer. And they were probably thinking they were maybe 2 years away from a potential clinical candidate. And I think by me joining, bringing some big pharma kind of thinking about, when is it good enough to move, we're able to move those programs forward relatively quickly and get them into the clinic within that 1.5, 2 years timeline. And that was the first time really getting these new model compounds into humans and showing that the preclinical data that we'd seen actually could translate into the clinic. And really the exciting part was everything we were doing was the first time it was being done. It's the first molecules made, the first ones that got to the healthcare, the health authorities approved to get into humans, the first clinical data, which was incredible data. And it was exciting just because people thought maybe these PROTACs wouldn't act like normal small molecules because they're larger than your average small molecule. And so they would have different properties. And so it was very exciting to see PK, PD data in the first initial number of patients. And of course, since then, I'm moving on to seeing efficacy data in a number of different programs as well. So those initial days were very exciting just because it was a first of every single time we were working on it. And now protein degradation as an area has grown quite a bit. There's a number of small companies, probably about 60 or 70, the last count, that are in the protein degradation space in general. And of course, the big pharma companies have protein degradation as one of their, I'd say, normal modalities now. So it really, over the last 8 years, 10 years, has really migrated into being another potential therapeutic approach that small and big companies take. And our challenge has been to stay as best we can in the lead in terms of innovation and moving our portfolio forward.
Yeah, absolutely. So whenever you look at the things that you're working on at Arvinas, what are some of the really strong breakthroughs that you're most proud of?
Yeah, so one of the clear initial breakthroughs was going back to this issue that PROTACs are, larger small molecules. They can range from 8 to well over 1,000 kilodaltons. So they don't naturally lend themselves to drug-like properties, the ones you'd like to see for a drug, in other words, the oral bioavailability. So the company's focus, the chemist's focus for the first few years was trying to find those kind of properties that could be imbued into a PROTAC that would make them more drug-like, ideally make them orally bioavailable. And the chemists largely did this by ignoring the kind of classic rule of 5, really trying to find a different set of rules for PROTACs, using biology in a different way, doing a lot more in vivo work well up front to see signs that could give chemists a lead into saying this particular ligand, this particular linker actually gives potential oral bioavailability. And over a few years, we cracked that as an issue to really show that PROTACs could be made readily orally bioavailable. And almost all the programs that we work on now have PROTACs that are orally bioavailable. And then we're able to segue that into understanding how to make them brain penetrant. And once we did that, that opened up the whole area of neuroscience, and particularly neurodegeneration, to us in terms of our portfolio. So those are the significant things. Taking that initial technology platform that Craig had basically invented and handed to us, and then adding different features like oral bioavailability, brain penetrance, understanding PK/PD relationships, then an array of different programs where you can see the proteins being targeted highly selectively for protein degradation. So a number of really quite significant firsts moving the technology forward. And of course, that has helped the rest of this kind of mini industry in protein degradation. So a lot of companies obviously know now how to make them orally bioavailable, and they're learning how to make them brain penetrant. And that's just added to the fact that the technology is, obviously ready for prime time as a therapeutic.
So I also read that you also have collaborations with other pharmaceutical companies. How have those partnerships contributed to your mission?
Yeah, great question. And when I joined, the company had already had a couple of deals done right at the very early beginnings of the company's history. A target, a deal with Merck, one with Genentech, and then when I joined, the Genentech one was expanded. We also did a deal with Pfizer on a target deal, and these were the companies handing us their interesting targets. We would find degraders and hand them back to them. So we weren't getting them into our portfolio, but they were going into those companies' portfolios. And the last deal we did of that type was with Bayer. And we also, at the same time, with Bayer Agrichem, set up a joint venture in the agrichem space. We made a company called Earth Bio, based in North Carolina, and we imported all of our technologies, and they've been focused on looking at how to deploy that technology into understanding disease control in plants and insect control. So basically showing you the ubiquitous nature of protein degradation mechanisms, and plants have many, many ligases, many more than humans, but the basic technology of making heterobifunctional molecules works in plants just as well as it does in humans. So yeah, there's been a kind of an interesting kind of migration of the technology over the last several years.
Very good. Can you share a little bit more, a little bit about your experience with both large pharmaceutical companies and smaller biotech firms, and what do you see as the key differences and challenges in leading these different types of organizations?
Yeah, so when we did the first deals, it was a great validation for the technology. So for any small biotech company, getting a deal done with a Merck or a Pfizer is just huge validation for the technology. You also get access to really smart scientists in the pharma company, and quite a lot of time you're getting access to their capabilities. So that helped Arvinas grow as we were working for these companies to find degraders for them, we were also learning and getting access and capabilities, which was incredibly helpful. And then we did a really significant deal with Pfizer on our major asset, vepdegestrant, where we did a 50-50 co-development, co-commercialization deal. And that is a very different, very, very different beast altogether. This is a co-sharing development, and for the last several years, where teams are working very closely together, sharing common tools, sharing common vision, really moving an asset forward. And what you're dealing with there is we're a small company working with a very, very large company. So things might change for us, but probably don't change as much as they do for Pfizer. So that is one of the challenges with a big pharma company. They do have this global footprint. They have many, many things going on simultaneously. And even though we've got this one very exciting asset, they have many. So keeping that intensity as being part of the kind of, I think one of the great things that Pfizer has done, keeping that focus on vepdegestrant and moving the asset forward, even though I know they've got many, many other things they could be doing. But it's worked very, very well. But it's a different scenario from just working on your own stuff. You're in those other deals, you were working for those companies to give them PROTACs that goes into their pipeline. And now you're in a co-development where you have to share and make joint decisions. But it's been a great learning experience. And we wouldn't have been able to develop vepdegestrant to the stage it's at, literally just waiting for pivotal data in the next few weeks and months without Pfizer.
Yeah, I'm less familiar with, I would say, the drug development collaborations that happen quite frequently, but much more familiar with collaborations for building things like personalized diagnostics and things like that in life sciences. And I always know that we would typically show up on calls and it'd be this level of anxiety because essentially you got the big brother and their smart scientists looking back at the things that you're doing, and there were always questions that they would oftentimes ask that frankly would carry us much further as well. I mean, so it does provide great benefits, and I could see that as well.
Yeah
for sure. So whenever I look at some of the things that you're currently working on with regards to cancer treatment, both with regards to breast and prostate cancer, can you discuss some of the potential impact of these therapies and what are the next steps for you?
Yeah, when we worked on androgen receptor and estrogen receptor, we knew there was good therapies out there for those diseases. But we also felt there's an opportunity for a degrader to offer potential differential pharmacology, differential biology. We believed in settings where there was high resistance to current drugs, a PROTAC might work differently. And the reason for that is inhibitors tend to bind to an active site of the receptor or an enzyme active site and have to sit there basically to make the effect occur. So it's occupancy-driven effects. Whereas with a degrader, it's not occupancy-driven, it's proximity-based. You're binding to two different proteins, a ligase and a target, and you're bringing them into close proximity to allow the ubiquitination process to occur, which is the cell's natural process. And when you get 4 ubiquitins transferred onto a target protein, it gets shuttled off to the proteasome and it's degraded. And then the unique thing is the PROTAC's released and it's allowed to do multiple different rounds of protein degradation. So a very different mechanism to an inhibitor. And Craig felt that would offer differential pharmacology and give a potential new therapeutic approach in areas where, as I said, you have either loss of expression of disease proteins or mutations to specific proteins that were causing disease. So that was the theory, and I think we've over the last several years we've shown that we can see that differential pharmacology both preclinically and clinically, and showing that a PROTAC, a degrader, and a proximity-based approach where you're hijacking the cell's natural system actually offers something different from just inhibition. And that's really where the excitement kicks in terms of looking at all the different potential targets. So vepdegestrant is our estrogen receptor degrader. We have bavdegalutamide and luxdegalutamide, which are androgen receptor degraders. And we just did a deal earlier this year to out-license luxdegalutamide to Novartis. So they'll be taking that compound forward into their pipeline, moving it forward into castration-sensitive prostate cancer and castration-resistant prostate cancer. So we're very excited about sharing the future of that asset with Novartis as it moves forward. But the technology itself, I think, now we believe is validated, not just by us, but by multiple other companies, showing that differential approach to a degradation versus inhibition, especially if you have a protein that's involved in scaffolding function. An example being something like KRAS, where there's multiple other proteins that are sitting in a complex. Yes, you can find an inhibitor that binds to that, but sometimes that protein's scaffolding function stays intact, and you can still see signaling come from that protein complex. With a degrader, you actually get rid of the protein, and the scaffolding breaks down. And so I think you get a more profound impact on signaling through that degradation. So some unique properties of degradation versus inhibition has allowed us to look at lots of different targets and really see its opportunities to differentiate, even with targets that have got a number of inhibitors already out there, either preclinically or in the clinic.
And how do you balance the need for scientific innovation with the business aspects of running a publicly traded company?
Yeah, well, yeah. So when I joined Arvinas, I was all about the innovation. I actually joined as the CSO. So I had no kind of real intention in my mind to become the CEO. But within a few months, I was also asked to become the CEO. But lucky for me, in a sense, that the training and background I got both at Glaxo and at BMS in terms of how innovation, invention can drive novelty and move things forward in a way that more traditional methods might not, allowed me to apply that in the setting at Arvinas as well. And we've hired people who are very focused on innovation. They don't just take the technology and let it sit. They adapt, they add to it, they really evolve it. And it's like any technology platform. If you don't evolve it and you just basically sit static, other companies overtake you. Your technology becomes obsolescent, and that's not something we want to be in a position to do. We are always trying to think of ways of innovating the whole space, looking at other kind of proximity-based approaches, looking at different ways of adding technologies to supplement the core technology. So there's a lot of that innovation going on. The aspect that for me is the business bit, which I was not prepared for, because that was not the job I was applying for, was all the interactions with investors and the fact that you've basically got to raise cash all the time. So, it took me a few months to recognize the fact that actually all you were doing was telling the story about Arvinas and telling the story about the technology and getting investors excited about the potential. Once that clicked with me, that it wasn't something unique, some new language that I had to speak, it was the same language I've been speaking for years. It's just that it was focused on investors and getting them excited. Then I was really comfortable with it. And of course, you in the biotech setting, you've always got to be out there getting investors comfortable that money is getting used appropriately, and that another tranche of money will also add to the value of the company. And I think it's something Arvinas has done very well over the last 7 or 8 years in terms of getting investor interest and moving our portfolio forward. But yeah, that was a big transition for me from purely just science to also suddenly, hey, you're doing the business side of it as well. It's been fun.
Yeah, I would imagine it could be, a little anxious in one way, but also validating in the other, right? So, I mean, just the ability to sort of see everyday people and talk about the things that you're doing in the language that, is intended for the kind of the target where these things are going to wind up, has also got to be validating as well.
No, it is the first few, let's say the first couple of years of talking about protein degradation to investors and external people, there was a lot of explaining about what the technology is and why it's different. Now we don't have to do that. Everybody knows the technology and they understand it, they get it. And it's all about how you're differentiating by the targets that you're choosing and the disease areas you're in. how good is your last data set, and how does it fit with the competition? So the dialogue on the platform has matured greatly over the last several years. But at the beginning, it was all about explaining the technology and why it was relevant. And I kind of miss those questions because we used to get, you could explain everything in a lot of detail, but now it's very much into the business. What's the data from your last data set, and what does it mean?
Yeah, very good. So that's a great lead in for my next question. What do you consider to be the biggest challenge facing the biotech industry today? So at a broad scale, and then how is Arvinas positioning itself to address those challenges?
Well, there's a number of kind of macro things that are conditions that have really impacted biotech over the last few years, which we're all aware of. I mean, clearly, COVID really set back the industry, even though the pharma industry was hugely important in developing vaccines and pushing back on the disease and helping humanity. There was a kind of a chilling effect on investment in the biotech world, and there was like global instability as well. And so I think there was a period of time, which we're still in, where the kind of free flow of money into ideas and concepts dried up somewhat, and investors were looking for more. When Arvinas was started out, it was very early preclinical data, platform data, and investors were putting money in because they were excited about the concept. I think it'd be tougher now to do that. I think investors want to see more validation. They want to get more comfort that things work. Ideally, if you're moving a program forward, they want to see clinical data more likely than not. In a way, in the past, getting investment preclinically was actually fairly easy. So the environment has changed over the last several years for biotech. It's tougher for biotechs to move forward. Now, I'm not saying there's not money flowing. There clearly is money there. It's just not as freely available. The number of IPOs really dried up considerably, compared to the time Arvinas did their IPO in 2018. So that's been one of the challenges. Although I think it does still drive innovation. I think innovation still gets access to money, still gets access to people saying that's something I want to invest in. So good data, innovative ideas, basically drugs that work, still bring in investors, still bring in the flow of cash that you need to develop. But for other companies, I think it's been tough the last few years, and hopefully that'll change over the next few years, and we get into an environment where investors are more comfortable again, going back into really truly speculative innovation. And these things are always a cycle, right? So we've been through a bit of a tough cycle the last few years, but I think hopefully we'll come out of that. Now, Arvinas was in a great position going through this because we just did a deal with Pfizer on our co-development. So we actually got a good tranche of money coming in. So we were in the lucky position not having to go out to raise cash all through the COVID years. We had a fair amount, and by the time we did need to do it, COVID was more or less over, and the environment had changed. But there's a lot of companies I think found the last few years pretty tough.
Yeah, I agree with you. I mean, I submitted a couple of different articles online that essentially said, look, as we were coming out of COVID, we definitely were experiencing a downturn in investment. A lot of the investors then were telling me, hey, look, in the next year, we're going to, start seeing things loosen up. By the time we got to May 2 years ago, things still hadn't sort of changed direction. And then, people were saying it coming into this year as well. Hey, look, we're seeing, again, another opportunity to kind of change the environment for investment. Heading into JP Morgan for next year, it does seem like there's a lot more investors talking about, sort of where they're going to start hedging their bets. So who knows? Maybe we start to see things open up a little bit more as we get into 2025. Yeah,
I think so. I think the beginning of the year, JP Morgan is also a pretty good indicator of where enthusiasm and excitement is. And usually a few months after that, you see potential deals appearing, the ones that have been worked on at JP Morgan. So I think we'll know relatively quickly into the year whether or not the environment has picked up, and the company either acquisitions, partnerships, or just in general IPOs to see whether or not we've turned a corner.
Absolutely. Last few questions are really focused on you. So what's the greatest piece of leadership advice that you've ever received?
Well, I mean, I do remember because somebody asked me a while back, I think it was a family member, why did you end up in science? And why did you stay in it? So there's a couple double answers there. So I get into science and get excited about science, mainly as a child watching the Apollo 11 landing. And I just couldn't believe this thing was happening in front of my eyes, that humans were actually flying to the moon and landing on it. And that captured my imagination. And I decided there and then I wanted to be a scientist. I didn't know if I was going to, I mean, obviously I wanted to be an astronaut, but, right. I got rid of that as an idea pretty quickly. But in general, I wanted to be a scientist. And I migrated into science courses and went to university. But then I was doing a degree in microbiology at Glasgow University, and the professor there in the final year, he also got the students into one-on-one interviews, basically to ask you, what are you going to do? What's your plan? And I remember him asking me, and looking back on it, I was probably a little bit prescient about what I wanted to do. I said, I wanted to get my degree. I wanted to do a PhD, maybe do a postdoc, maybe join a pharma company or a biotech company. That's the trajectory. And he was listening to me, he just looked at me and says, John, you better have a plan B, because that's not going to happen. So his kind of not disgust, but his thoughts of like, that is way too linear. That's not going to happen. You better have a plan B. So I not necessarily had picked up a plan B, but I did think about other things I should be doing. But as I was doing that, I still went through that same trajectory. Did my PhD, did a postdoc, joined a big pharma company, joined another one, then joined a biotech company. So I do look back on his kind of, incredulity that I would have a clear path laid out for myself and say, have a plan B. And I did work up different ideas, but stuck to the path.
Yeah, I don't think that many people have this linear sort of path anyway. So most paths are very wavy through time. And then, when you're presented with opportunities, you have decisions to make, right? So
exactly.
Yeah. What inspires you?
Well, I think I am inspired by science and innovation. When I look at the things that get me excited, it's reading about scientific breakthroughs, and it does go back to when I was a kid, seeing things that happen are like almost fantastical. But in the kind of the health sciences area, all the kind of breakthroughs in terms of drugs that can really impact disease in a really positive way. Operations and surgeries that are almost miraculous. That ability in the kind of the biology medicine area to really change human life in a positive way, I have always found like amazing. And just to be part of it in a small way is incredibly inspiring for me. But more broadly, just technology in general, I'm always amazed at the things that when I look back at when my dad and my mother were around, when they were born. My dad was born in 1916, and there wasn't a lot of technology around in those days. And thinking about how as he came up as a child through his teen years, apart from having to go into war, the technology development and things that we just take for granted, that generation didn't have access to, and we just accept computers, phones, cars, everything, and it's all technology innovation that has come over through the decades. And I think that applies to medicine. I think we're just at the beginning of, I think, a whole new frontier of medicines that can have a huge impact in human health. And that's always, so technology, innovation has always got me every single time.
Absolutely. What concerns you?
Well, I mean, on the other side of that, what concerns me is that it gets taken for granted, right? I do worry about science, innovation, all the things that we have getting taken for granted because it all looks too easy. There's the kind of an illusion that somehow these things just fall off a log, and then you have a new medicine, a new technology, a new thing. And we all know, the ones that work in the industry, it's incredibly difficult. And it's a huge amount of failure associated with innovation. And for every drug it makes, there's 100 that don't. And there's scientists that can spend their whole career working in a pharma company that never experience a drug getting all the way through to approval. And so for me, that's a concern that gets embedded to a certain extent, both in terms of culture and politics, that innovation, medicine, innovation in general doesn't get rewarded. And there's a few that, it's easy. And it's not. It's one of the most difficult things we could do.
It's one of the biggest struggles that I have from the sidelines is this whole idea of, medicines being too expensive, which in part I agree with, but there's a large part of me that also says, well, hey, look, when we went through COVID, thank goodness that there were all these companies doing research on, SARS and MERS and Ebola and all these things that essentially impacted how we might approach solving the problem of vaccinating against COVID. So,
exactly.
I don't know.
I think the mRNA technology as well that companies were working on. It was the perfect juxtaposition of previous viruses that had occurred and a technology platform that could be applied to it. Amazing.
Yeah, absolutely. Last question. What excites you?
Yeah, I mean, what excites me is always the next thing for me is that could be going to a nice restaurant or a movie, or I'm always excited about what's coming next. I always like to have a plan ahead of me. My wife knows that a lot, so she's great at coming up with trips that should be planned and people we should see. So on the personal side, it's always about the next thing we're going to do and enjoy. On the business side, clearly, for Arvinas, we've got some big stuff coming up in the next few months. This is the first pivotal data for this company coming out at the beginning of the new year with vepdegestrant in second-line metastatic breast cancer. So it's exciting for the company. It'll also be an important point for the technology for everybody. This will be the first PROTAC that gets pivotal data. And hopefully the data will show that it's an approvable drug. We'll see what the data tells us. And if that's the case, then we'll be in a position where we get to launch the first PROTAC into that space. So that's a major event for the company and for the technology. And if positive, I think it validates the rest of our portfolio and to be quite honest, other companies' portfolios in saying these technologies work and really add benefit. So the next thing is let's see what the data says and let's see what comes after that.
Exciting for sure. Well, John Houston, thank you so much for being on the Life Science Success podcast. I thank you so much for being with me and answering all these questions about Arvinas and your life as well. I mean, it's great to hear your overall story too, and I appreciate it.
Thanks so much, Don. Really, really enjoyed it. Thank you.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it. And last but not least, rate the podcast. Thank you again.